Now, in this video or section, we're going to talk about cellular immunity. So we talked about humoral immunity, which is B cells. Cellular immunity is going, or cell-mediated immunity is going to be T cells or T lymphocytes. Both humoral and cellular, we kind of have three stages. Both recognize their antigen, so they'll be able to decipher their specific match. So same with B cells. There's going to be a T cell that has its specific antigen that it needs to find to recognize to then also start working. They attack. So B cells are going to attack with antibodies. T cells are going to attack a different way, and we'll talk about it. And they both have memory. So T cells will make memory cells as well to remember this same antigen's been around once, so I'm ready for it the next time. And same with B cells. So you can think of it as recognize, react, and then remember. So you recognize its specific antigen, have a reaction of some sort, and then can remember it for later and can have a faster reaction the next time. So we talked about MHC proteins in a previous video. Just as a review, MHC1 proteins, those receptors, are on all nucleated cells, so not red blood cells. And they are showing endogenous antigens. What does that mean? It's going to be antigens that are intercellular. So again, MHC1 is going to be important in showing that these cells are infected. So again, if an antigen comes inside of a cell, then what the cell will do is present a piece of that antigen on its surface, on its MHC1 receptors or proteins, and it'll show it. So this usually is saying, hey, it's me. So usually it has a self antigen, meaning it's saying it's a normal, it's me, I'm a normal cell. So they won't be attacked. But if they're infected, they're going to show, hey, I am compromised, I need to be destroyed. So if they're infected or abnormal, meaning a cancerous cell, then they're going to show non-self proteins. So they're going to show those abnormal proteins, and they'll activate what we call cytotoxic T cells, which are also known as killer T cells. So you're going to see killer T cells as kind of, or cytotoxic T cells are going to be the ones that'll recognize that this is abnormal, and it's going to take that cell out because we know it's infected and there's no saving it. MHC2 proteins are again on antigen presenting cell, and this is showing you exogenous antigens, meaning it's going to be ones that are extracellular that are then phagocytized and taken inside the cell, and they're processed and shown by those antigen presenting cells. So they'll have kind of showing some sort of antigen. So remember the types of those, that they're macrophages, dendritic cells that are in connective tissue in the epidermis, dendritic, and then B cells themselves. Because remember, they take in the receptor antigen complexes. So these are going to be presented on MHC2. So only these cells have MHC2, and they're recognized by helper T cells. And we're going to talk about helper T cells, that there again is the party coordinator. So they help coordinate the immune system. Again, antigen presenting cells are nucleated cells. If they are nucleated cells, then they still have MHC1. So if there is a pathogen that's going to impact these cells, then they will show antigen on MHC1, or if they need to, they'll present to both killer or cytotoxic T cells and helper T cells. So these are helper T cells. These are cytotoxic. So again, antigen recognition. If you have an inactive T cell, it's going to come along and it's going to recognize the antigen that's presented in its MHC proteins. So this is looking at MHC1 because this is an infected body cell. So again, it's going to be intracellular. And again, antigen presenting cells have both. So they recognize MHC when they have their specific antigen. So again, it's only going to be specific. This T cell recognizes only this antigen right here. If it was a different shape, say if it was a square, we'd have a different T cell recognizing this shape. So we have two types or classes of T cells. We have ones that are CD8 that will develop into cytotoxic or regulatory. We'll talk about regulatory a little bit. And they, again, are responding to MHC1 proteins. So they're responding to the ones that are on all nucleated cells and going to be intracellular pathogens that are presented on the MHC1. CD4 cells or markers, CD4 T cells are going to lead into helper T cells when they're activated, and they're responding to MHC2 receptors, which are going to be on your antigen presenting cells. So again, B cells, macrophages, and dendritic cells are going to be your major antigen presenting cells. So oftentimes this can be intracellular, but also it's going to be extracellular because these are bringing them inside the cell and then presenting it to on the MHC2 to helper T cells. So when a CD8 T cell encounters its antigen, and again, it's a specific antigen, to class 1 MHC protein on the surface of the cell. So again, these are going to lead into cytotoxic T cells. So what happens is you have an inactive cell. Because you want them inactivated, especially cytotoxic cells, they're inactivated, they meet their antigen, it's presented to them. So this is the infected cell. The virus or the bacterial antigen is presented on MHC1 here. This T cell is matched for this antigen that's being presented. So it comes in and it matches with it. And then we're going to see activation, and these are going to be active cytotoxic T cells. And we're going to see the same thing, that we start making a clone army, and then memory cells as well. So we're going to see that these are from CD8 cells. Now they're activated and they're cytotoxic T cells. With this activation of the CD8 cell to a cytotoxic T cell, the T cell has to have what we call co-stimulation. So this can be either another receptor or another kind of pressure receptor or some sort of thing that's going to help activate the T cell as well as the MHC1. So you have to have the matching MHC1 with this antigen. And you have to be able to co-stimulate to activate the CD8 T cell. So we think of it as T cells have kind of two ways that they're activated, because we don't want cytotoxic T cells to be activated inappropriately, because then they're going to start killing kind of normal cells, and that's not something that we want. So you can think of it as a safety on a gun. So you have a loaded gun, it's ready to go. To have co-stimulation, you have the safety on, so you have to take off the safety for you to be able to fire the gun. So it's the same way. It has to have both the antigen that it sees and the co-stimulation that it gets to actually activate it into a cytotoxic T cell where it start to kill cells. So there are three types of T cells that can fight against the same antigen. So these again are CD8 cells. We also have another type of cell, which is the CD4, which we'll talk about too, which is the helper T. So cytotoxic T cells again are your killer T cells. Then just like when we saw with the B cells, we have memory T cells, so we can have a faster reaction if we see that antigen again. We also have regulatory T cells, which are going to help regulate the reaction. So we'll talk about those next, or in a little bit. So first, cytotoxic T cells are going to be the effector cells. They are going to attack and kill the cells, because again, if you're saying that there is something inside the cell that's infected with it, the cell will wave its flag on the MHC1 protein, MHC1 receptor, and then CD4 or cytotoxic T cells are going, not CD4, CD8, cytotoxic T cells are come in and kill this cell, because there's no saving it. It's just better to say goodbye and cut your losses. So the target cells is either a virus that's inside the cell, intracellular bacteria, intracellular parasites, cancerous cells, another one, and cytotoxic T cells are the reason why transfusions, or if we kind of have a mismatch, or transplants majorly are going to fail. So why do you take immunosuppressants if there's a transplant? A lot of times it's going to be because of cytotoxic T cells. They're going to recognize this transplanted cells, whether it's a kidney. It's going to have abnormal MHC1 proteins because it's not the same as yours. And it's going to recognize it as foreign, and it's going to kill those cells. So we need to make sure that the cytotoxic T cells do not do this. So when we see it recognizes its antigen on the MHC1 protein, then it kills like we saw with natural killer cells. Remember, those are a non-phagocytic, non-specific. So they're going to kill in the same way, but this is because of a specific antigen for cytotoxic T cells. So first they release perforins that's going to pop a hole or poke a hole in the plasma membrane. So first off, we're going to have a hole in the plasma membrane, so then you can't regulate water flow. And then granzymes are going to enter and continue to cause cell death. You can also have, if this is fighting against a virus, then interferons are going to be released. Interferons, remember, go to neighboring cells and cause them to make sure they're ready for any virus infection. So they'll stop viral replication or viral reproduction. So then they aren't making new viruses, so then new viruses can't spread around. And they can also increase the activity and bring in macrophages, our big phagocytic cells. They also release what we call tumor necrosis factor. That's going to, again, bring in the macrophage and help kill any cancer cells. So if we recognize that it's a cell that has uncontrollable growth or division, then we need to take care of it. And so the cytotoxic T cells will do so. So it releases, and then it can move on to the next cell. So again, if there's in a line of cells are infected by our intracellular pathogen or intracellular antigen, then these cells all are going to be presenting on MHC1, and then these cytotoxic T cells are just going to come in, poke a hole, release the granzyme, kill it. Poke a hole, release the granzymes, kill it. And so they'll just keep going. Remember CD8 memory cells? We're also going to have CD4 memory cells. So both helper T cells and cytotoxic T cells are going to make memory. So they're produced, but again, they don't differentiate into cytotoxic T cells or regulatory cells until the next time it meets its match or its antigen. So on second exposure, it's going to become more memory cells, and mainly memory cells immediately are going to be ready. Memory cells are going to be ready, and then they'll just immediately go into cytotoxic T cells, and then they'll be able to kill. So again, faster response when we have memory. The other type of CD8 cells, remember, it can go into cytotoxic T cells or regulatory T cells. These are going to be actually immunosuppressor cells. So they are going to decrease the response. They only start after the first antigen exposure, the initial first immune response. Because we don't want to necessarily stop the T cells or the B cells initially. But again, if you're exposed to the antigen and you have a lot of cytotoxic T cells around, well, this is going to be a way that we can decrease the response. Because we don't want cytotoxic T cells. Again, if there's just a line and they keep going and killing cell and killing cell and killing cell, well, the regulatory T cells are more like our stop signs. So they're saying, stop killing, you're done, we're good. So we need some way for the T cells to just stop and not continuing to respond. And that's regulatory T cells. So again, we have cytotoxic T cells. They are going to respond to MHC1 and have co-stimulation. We also have memory cytotoxic T cells to remember if we have a future same antigen in the future. And then we can have delayed suppression. Again, eventually the regulatory T cells are going to say stop, slow down. So again, the cytotoxic T cells are going to release perforins. That's going to destroy the membrane, poke a hole in the plasma membrane. It's going to release granzymes and also cause cell death, apoptosis. And you can also see where it's going to stop, another way is lymphotoxin release, where it's going to stop the metabolism of the cell or chemical reactions in the cell. CD4 are helper T cells. So antigen presenting cells are going to be stimulating these helper T cells. So CD4 T cells are going to produce helper T cells. These are going to help, again, kind of party planner. They're going to activate B cells. So what can happen, remember, a B cell is an antigen presenting cell to helper T cells. So they can kind of keep talking to each other and help each other out. So again, that's why they're helper T cells. They respond to MHC2. So helper T cells is 2, cytotoxic T cells is 1. There's still co-stimulation that you need. So you still need kind of that co-stimulation factor to activate. And like I said, you can have helper T cells, which are your active ones that are going to help all out. Or you can have memory cells to remember for future use when you meet that antigen again, if you do. So helper T cells, they're kind of in the name. Again, they're like the party planner. So they'll coordinate and activate pieces of the humoral and cellular immunity. Again, we're going to the point where one antigen, one bacteria, is going to stimulate all of your immune system. We're separating it into different parts, but we need all the parts to fight against an antigen properly. So once activated, again, antigen presenting cells, the macrophages, the dendritic cells, and the helper T or the B cells. Once the helper T cells are produced and activated, they can activate more B cells and other T cells. So they can send out signals. I'm not going into all the chemicals that are involved, but basically helper T cells are going to help send out signals to help the B cells be more active and T cells to fight against this pathogen. Again, these are specific, so they're going to say the help sign to the flu virus, a specific strain. They also help with T and B cell proliferation. So again, you have an activated B cell or an activated T cell. The first thing that these guys are going to want to do is make an army. So they're going to proliferate, meaning they're going to divide and make a clone army. So without helper T cells, we're not going to have a clone army. They need help with this. They also produce what we call cytokines, which are going to be chemical messengers to help bring in other immune cells. So it's another way we communicate. So we're saying B cells are activated and proliferating, T cells are coming in. Well, now we need neutrophils and more of our non-specific innate immunity. So these are going to be our second line of defense. Our cells, our non-specific ones, are going to come in to help. So again, we have all arms of our immune system that are going to come and help to that area against that pathogen. The neutrophils and the natural killer cells and the macrophages don't care what antigen it is. These guys are specifically recognizing that antigen. And a B cell can recognize the antigen, and a T cell can recognize the MHC protein presenting that antigen. So we can have, again, multiple parts that are all activated. Without our party planners, without kind of our helper T cells that have their hand in everywhere, they have their hand in the innate or the non-specific, they have their hand in the humoral, they have their hand in the cell mediated, they have their hand everywhere. So without our party planners of making sure that everything's kind of coordinated and helping each other, there's no immune response. So you get rid of the helper T cells, then you get rid of the immune kind of immunity, your immune system. So this is just showing you again helper T cell. So this is an antigen presenting cell. It's actually a macrophage, but it can also be a B cell or dendritic cell. Is showing MHC2. It's showing an antigen that this helper T cell recognizes, and then this helper T cell is just going to send out communication everywhere. So it's going to kind of don't need to remember all of the names, but it's going to send out chemical messengers to macrophages to increase their activity, bring in more white blood cells, inflammation. So it's going to help kind of get the non-specific defense involved. It's going to send out information to B cells to make clones of themselves. And so then it's going to be involved in the humoral. Then it's going to clones of cytotoxic T cells. And so it's involved in cellular immunity. So it's just going to send out messengers everywhere. Messenger, messenger, messenger. So this is just looking again at the combination of kind of how everything works together. We again divide it into different arms, but they're all simultaneously doing things at the same time to fight against the same antigen. So you have your non-specific defenses. Remember, you have complement, which are going to have the membrane attack complex and help kill and also coordinates the immune reaction. You have natural killer cells and macrophages that are going to either phagocytize, or natural killer cells are going to attack like cytotoxic T cells. You're going to have activation of your T cells, cytotoxic T cells, of helper T cells that are going to help everywhere. You're going to activate B cells that are going to make antibodies from plasma cells, produce antibodies that'll fight. So it's just kind of everywhere is going to help fight, just in their different ways. So they just have different ways of fighting, but they're all the same goal, is to get rid of this antigen. So bacteria, oftentimes a macrophage is going to come in and consume them. That's an antigen presenting cell. Same thing, activation of helper T cells, which will help with activation of B cells further. Plasma cells make antibodies, and you cause this bacteria to be destroyed. And then you activate cytotoxic T cells that destroy the bacteria directly. Viruses, same thing. So you can have infection of those phagocytes or they're eating it. You have antigen presentation. So again, that'll be on MHC2. Activates helper T cells. Same thing. B cells activated. Plasma cells make antibodies, and you destroy the viruses or stop them from getting into cells. Or you have the infection of the cell. Antigen is going to be presented on MHC1 by those infected cells. They're going to activate cytotoxic T cells, which will kill the cells, or natural killer cells will come along and recognize it as foreign, not specific. It doesn't matter what type of virus it is to natural killer cells, because remember, they're non-specific, but they'll kill in the same way. You also have the interferon release, so that you're going to stop viral spread.